Hello. So, in this class we will be talking about some basic aspects of bioinorganic chemistry and which will be covered with in this first class particularly introduction, which will deal with some of the basic aspects what little bit we know, and some more will be covering in this class. And related to the second one is the properties of biological molecules. And these properties are basically different from what we know about the typical molecules, where we have some thing where we know that the basic skeleton is made up of say your carbon, hydrogen, nitrogen and all this, and most of the time we will be talking about, since the inorganic part of this will be covering as starting with the metal ion. Suppose we have the hemoglobin where we have the iron, and that metal part, if we just simply consider, and if we consider that metal is coordinating with the some of your important ligands, we can consider this as the biocoordination chemistry in that way. So, how these properties of all these biological molecules are different from that of our simple typical small molecules, where you have one metal ion and several ligands. So, if the source is different, that means if the metal source is all the biological source and the ligands are all biological source, and when they are interacting and how they are following, whether they are completely different from what we know from our laboratory knowledge, that how they are giving some metal complex and how they are forming in our life processes. So, the next one will just cover that choice, uptake and assembly of metal containing units in biology. Where we will see that how the nature has a typical choice process such that it can take the particular metal ion or some non-metal species for its typical action, and how it can take for its utilization, and finally, the assembly of metal containing units. Assembly is a typical process where we can have, suppose the metal center is there, and when it is coordinating to the biological ligands or biogenic ligands, where the source is biological only, and that is forming some unit what we call as the metal ligand assembly. So, you have one metal, and depending upon its demand or coordination environment or the nature of the metal center, it can bind either 2 ligand, or it can bind 4 ligand, or it can bind 6 ligands together around the metal center. But this particular assembly process, what we will be talking there, when we have one particular metal center and a mononuclear unit is functioning for some important reactions. Suppose we know a little bit about that hemoglobin or myoglobin system, where we consider that the iron center is there and some of the donor groups are there surrounding the metal center and forming a mononuclear unit. But if we go beyond that, that means the mononuclear system is giving through some bridging ligand to a binuclear system, or that binuclear unit is further taking up another center giving you a trinuclear unit. Because this is important to know, where we know that the assembly of metal containing units, how we can have, because we are utilizing this metal salt, say iron, from our body, but we are taking this particular iron from our dietary sources, from our food material, and that iron is going to our body and we are slowly absorbing those iron. And how this is basically related to our laboratory knowledge, that where we use some metal, we take some from reagent bottle, you have the ligand already synthesized, but for in our body or in plant system or in any other living system, we do not have all these things. So, we should have the source of the metal as well as the source of the ligand. So, how we get all these metal centers? Metal centers we are getting from the food material. So, this uptake is important in that way, how the system is taking up that iron, and that iron will be utilized for some useful purpose. Suppose that iron is utilized for our blood synthesis or hemoglobin synthesis, but for that particular synthesis what we need? We need the metal as well as some ligand, say porphyrin ligand, but you should have the source for the porphyrin as well as the source for the iron. But you cannot wait till you reach the corresponding available iron site. That means, you have to wait for food material what we will be taking, or iron source, direct iron source we are taking sometime as some iron tonics or something. But you have to wait for the hemoglobin synthesis or all these things, but the hemoglobin synthesis is a regular process. Throughout the day and throughout the month or throughout the years, we are continuously synthesizing that thing. So, we should have some regular source, and that regular source we will deal with afterward, that what should be your iron storehouse, how you can store iron in our body. So, for that purpose also we should have some specific mechanism where we can absorb iron from your food material or any other source, and that iron is taken up by some small ligands or some groups, and that is there, and it is transferred from one particular part to the other part, and that part is responsible for storing that iron. That means, the storehouse of the iron. So, that is why this particular the assembly process is important, because in that assembly process we will store large number of iron atoms. So, the next one which we will be talking about is the therefore, your metal storage. Then transport and biomineralization. So, you can store the metal centers. It is not only true for iron, it can be true for zinc, it can be true for copper, because only 3-4 metal ions we know their concentration is very high in our body, and those metal ions are useful. So, we should have some storehouse, but only what we know so far about iron only, but we do not know in detail that how the zinc is stored in our body or copper is stored in our body. But in the recent developments or the recent studies what we are studying nowadays, because throughout your life, as we are going older and older, suppose after 70 years or after 80 years or after 60 years, how these storehouse of these metal ions are behaving, are all them are utilized for your blood synthesis, or they are doing some degenerative processes in our body. That people are talking, people have started inquiring all these things, how these two diseases we will talk about little bit afterward, the Alzheimer's disease and Parkinson's disease, whether those two diseases are related to the accumulation of your metal ions in your brain. So, not only that, when you take the food and from the stomach the metal ions are accumulating, then slowly through your serum or the bloodstream it is moving through your entire body, and ultimately little bit of that for our neurological processes or generating the neurotransmitters is going to our brain also. So, for that particular purpose what we see that metal storage, transport and biomineralization. We know all how nature is doing for us, how nature is storing the metal ions in a ore or mineral fashion. This is the same thing, the biomineralization term we will be discussing afterwards, that how the biological system is also responsible for some type of that mineralization process. We know iron in the nature, we know that they are available in the form of hematite, magnetite and all other iron ore. From your school days you are learning all these things, but in our body how they are getting stored from the free supply of the ions. The supply of those ions are in the Fe2 plus or Fe3 plus, and those are then stored in our body in some typical form, and that particular form will considered as your biomineral. Next, we will just talk about the atom and group transfer. Chemistry. This is nothing but how we can transfer one particular atom or a particular group from one system to the other. The way we know that the iron center is responsible for accepting our dioxygen molecule when we get the oxyhemoglobin or oxymyoglobin molecule. So, the entire thing is that here is dioxygen molecule which is getting attached to your metal center, and dioxygen is behaving as a ligand. But in this particular case, what we see that the atom and group transfer. So, in the biological systems, if we find that the iron center is there, and that iron center, if we have that particular iron center which is attached to the dioxygen molecule, and that dioxygen molecule afterwards, if you can find some mechanism is there, and it is happening in our living system also, that if you have a oxygen cleavage, we know there is a very strong bond there, that oxygen double bond is there, but if you are able to cleave that such that you can make something where if you have the FeO system, the ferryle system or the FeOxo system, which is very much similar to that of our typical some other metal centers, what we all know that the vanadium system or the molybdenum system, and how these oxygens are transferred, because this oxygen is utilized for making some important transformations or making some important molecules, that this oxygen, whether that particular oxygen, say for some example that that corresponding reaction, what we know that the insertion of the oxygen atom say in some CH bond, what we call as a oxygen insertion reaction, and for that oxygen insertion reaction what we use? We use some paracids or some reagent, so metachloroperbenzoic acid or iodictyl benzene. So, that oxygen is getting transferred to some of all these organometallic type of compounds. But if in the biological system that is also required, that we have to transfer that oxygen from one system to another, and that particular thing is basically activated by the metal center. So, metal oxo system. So, most of these cases we will talking about the metal, not nitrogen or sulfur or other case. So, we will talk little bit about this metal oxygen system and how we can accept that oxygen. That means, you have the pure system where you have the metal center, it is taking up this oxygen. So, you are getting that particular oxo complex, and afterward that oxo complex is transferring that oxygen for oxidizing some other species. Next, we will be talking about that electron transfer proteins. So, all these things, because we are talking about iron. So, why iron is so important? Because we know much about the iron center, starting from your blood, your cytochromes and all other system, and some of these metal centers, when we consider that this is a basically a consideration for the centers where you have the metal centers which are either redox active or redox inactive, like zinc and iron. So, some of these reactions, some of these characterizations or some of these properties will be typically related to zinc, where you cannot get some electron or you cannot put some electron to the system. But if it is a system which is iron based, and we all know that iron is settling very easily between its +2 oxidation state and the +3 oxidation state. So, you can take off large number of electrons from the system, or you can put large number of electrons to that particular system, because which is very much required when we try to oxidize or reduce a particular system. Suppose we try to reduce the dioxygen molecule for our assimilation, for our food oxidation, we try to reduce dioxygen molecule to water. So, we have to supply 4 electrons. In the same fashion, in the photosynthesis also, we try to oxidize the water molecule to get the dioxygen molecule. So, for all these electron transfer reactions and the molecules which are involved in all these redox processes, we will be talking about this electron transfer, but they are from protein origin. So, biological molecules which can supply electron and which can take up electron. So, they are oxidoreductases. So, they can provide for your oxidation reactions and they can also provide your reduction reactions. Then towards the end of this course, we will be basically talking about metal nucleic acid interactions, where we will see that we all know that the component for the DNA, RNA and all these, the nucleic acids, the nucleotides and the nucleosides and all, but how they are interacting with the metal, whether the metal has some role to play for the different reactions related to your DNA and the RNA molecules. And lastly, we will talk about the metals in medicine, which is very important, because some of these metal containing important compounds we can use as medicine. But before that, we should know a little bit about something where the metal center is directly involved for some of our diseases. So, then only we can find whether it is your cisplatin is an anti-cancer molecule or not. How we know that? That means, if we know that the function of this particular platinum complexes and all these reactions, how it can help, that the metal center is required and 2 of these donor groups are coming from your DNA molecule or the any other living molecules, and they are doing something where you can stop the propagation of the tumor growth or any other growth system, or whether those metal centers can be involved, whether you are using a redox active metal center or a non-redox metal center, that for the DNA cleavage reactions. For that, what you need, that you have now the DNA, we can consider that DNA as a very big ligand molecule, because we know all the donor groups are available and metal is very useful to bind. We all know that is all the time, if nothing is available to the metal center, it can bind to water molecule. So, it can bind to water molecule, and that water molecule after deprotonation, it can have a corresponding hydroxide complex. So, in that particular form, that whether nothing is available, you can have a metal center, which that means the availability of the metal center close to your all these nucleic acids or the medicinal molecules is important. But if that particular water molecule can be substituted by some other donor groups from the medicine or from the nucleic acid, so metal will all be interacting with all these useful groups. So, that we will see how the metals are interacting with nucleic acid as well as the medicine. So, basically some useful books we will just cover or use the study. You can have those books. Number 1 is your bioinorganic chemistry by Pertini, Gray, Lippard and Valentine. Second one is Principles of Bioinorganic Chemistry by same Lippard and Bark. third one is biological inorganic chemistry. By Crichton. And last one is also related to this and is a very handy book, it's biocoordination chemistry. By Fenton. Okay. So, the basic classification for all these molecules, that means whether you can have a typical metallobiomolecule and these metallobiomolecules, you have the metal center, and obviously we will be talking about those metal centers, and we will always refer wherever you go and wherever you study all these things, that how these metallobiomolecules are classified depending upon their function. So, there are certain aspects that metallobiomolecules you can classify as per their functional behavior, as their metal ion content, because when we look from a inorganic chemist's point of view or a coordination chemist's point of view, how you see a particular metallobiomolecule. A biologist can see from his biological side with eyes, well trained, well expertised eyes of biology. But when I see or you see that from a coordination chemist side, that means you will first look at the metal center and will always try to gather informations related to that metal center, and that is why you always interested to those molecules only. We are not talking any system where you have a system, some enzymes and some protein molecules where no such metal center is there. But these groups like these metallobiomolecules, suppose you have some metalloenzymes. Why we are talking all these things? Because we know the property of these metal ions, we know how they are coordinating to the water center if you have the water as the ligand, or if you have the ammonia as the ligand, or any other small groups as the ligands. Because we know how this metal can interacting, because the bonding patterns, the detailed chemical pattern or the chemistry pattern, chemical bonding is known with respect to that particular metal center. So, for us, for this particular type of class, it is very easy to classify all these with respect to the metal center. If I ask you that how many of these metallobiomolecules you know related to that particular metal center, say iron. Next, we should know what are the different structures, because when you have the iron site in a test tube in a laboratory class, if you say iron in your test tube, and then you put the ligand, any ligand starting from your water, the most simplest possible ligand is the water molecule, to ammonia or any other complex ligand system. You immediately tell me, no, this is forming a corresponding octahedral compound or a tetrahedral compound or something which is a dinuclear compound. But all these cases you do not know the detailed structure. So, once you identify the metal center, next thing what we try to know is the corresponding structure, because the structure is all the time very important when you go for some complex ligand system. So, we will see that your ligand is not a simple ligand now, your ligand is not water molecule, your ligand is not ammonia molecule simple, but they are complex. So, when you have a complex ligand system and you put that metal on that system, what is happening? The metal will always try to find its available donor sites. Suppose iron is there and you having a complex ligand system. So, iron will try to coordinate to the available donor atoms, whether it is nitrogen or oxygen or sulfur. So, the organic part, that organic complex molecule of biological origin, will have to find out that how many oxygen are there, how many nitrogen are there, or how many sulfur groups are there. So, immediately those groups will come close to iron, and if they are falling at a bonding distance of 2 angstrom or so, they will try to form some bonds. But if your available donor groups are less than those required numbers, say we are looking for a octahedral complex for iron, but the available groups are less, that means available groups are 4 or available groups are 3, what will happen? Your iron will bound to that particular organic ligand, and you will have some vacant positions, and those vacant positions are very useful, because in the laboratory you cannot control all these things. Once you dissolve any iron salt in water in a test tube, what will you find? That you are getting a hexaco complex of iron 2 or iron 3. You cannot say that okay, I have something, that means only 3 water molecules are coordinated to the metal center and remaining 3 are not there. But in the biological system, to know the structure, you will find that those 3 groups from the that organic part of the molecule is coming and attaching to the iron center, and you are having 3 vacant sites. If it is in the water system, because most of the time we will be talking about water, in our living system it is water system. So, they are all will be coordinated by water molecule. So, the next thing, once you identify the metal center, next thing you should look at the corresponding structures, and for that metal is playing an important role. All the time what we are talking so much about all these metal enzymes, because protein crystallography is a huge subject, is a vast subject, and that protein crystallography, when we try to get the corresponding crystals, so we use X-ray to do for the structure determination, and for that structure determination we always see that some X-ray is used and the diffraction patterns we are getting. But the first thing that you should have the right sample, and that right sample is in the crystallized form. So, all these biological samples devoid of metal center, they are all of biological origin, so typical organic molecules, and sometimes those organic molecules are very big, they are trapping huge amount of water molecules or some other molecules. So, it is very difficult to crystallize those molecules for X-ray diffraction. But if it is a metal enzyme or metal containing molecule, so that particular system, that with that assembly of the metal and the biological ligand, it is easy to crystallize. And during that structure determination, it is also easy to identify without taking the help of other spectroscopic properties or the spectroscopic determinations for the metal ligand distances or the metal ligand size. You can find out or you can locate from the diffraction pattern precisely the metal center. The first thing what we can do, because the atomic mass is very high for the metal, so that particular technique is known as the heavy atom method. You can locate the metal precisely and very accurately. So, that is why it is giving you a useful structure. If you do not know the detailed structure, suppose the detailed structure of your hemoglobin molecule or the myoglobin molecule, but if you have a diffraction pattern and which you can partly solve, you can find out or you can determine the structure which is your immediate environment of the iron, and that gives us some idea that we can find out the immediate environment of the iron, and which is also very important to know the function of the metal related to your function of the hemoglobin or the function of the myoglobin. So, 3 things we can together we know to identify the metal center, then you know the structure, and then how this particular structure is related to your functions, different functions. So, these metal or biomolecules, so there are typically we all know that one part you have the protein part and another is the non-protein part, and in the protein part you can have the enzymes when you have the metal center, they are all your metal enzymes, and these protein parts can be further subdivided into two, and the other part is your transport and storage. And this transport, because we will focus our attention here only, because we are not elaborating the entire subclass of all these things, because if you know all this classification initially, you can tell what particular type of metal or biomolecule we are dealing with, and how these metal or biomolecules are related to some of the names of these molecules. So, if you have this transport and storage in terms of electrons, they are basically electron carriers. That means, you can have large amount of electron, and these protein molecules which are important for this, that means they are basically your metal or biomolecules, and those metal or biomolecules are protein in nature, and they can be useful for transport and storage of electrons. So, these are basically electron transfer molecules, and those molecules we all know, they are the examples are your cytochromes. So, what are cytochromes? From this chart or this table, you can have that what are cytochromes, you can be able to tell that they are electron carriers, they are a transport and storage type, because they can tunnel electron, they can store electron, and they are of protein nature, and they are of metal or biomolecules type. So, in these cytochromes as well as your iron sulfur proteins. So, when we identify, what I was telling you, that when you identify these centers, that what are your iron sulfur proteins, that means when your name is there, you can immediately tell that is iron is there and sulfur is there, but the metal center is present is your iron. But for the cytochromes also, you have the iron center present there. And now, the next one is important, and we will start from there basically, which is your metal storage and structural. So, how we can utilize these protein molecules to trap your metal ions, and those trapped metal ions can be stored. So, you will have a storehouse of these metal ions, and this storing of these metal ions can also be utilized for your different structural pattern also. So, under this category, we will consider two of these molecules. One is your ferritin. So, these ferritin molecules are basically your metal storage system, where we can store our metal ions. In our body also, we store these metal ions as ferritin molecule. So, how you get these ferritin molecules? So, ferritin molecules, you will have a huge protein envelope, and within that protein envelope you can store large amount of iron centers. So, basically this particular, if the detailed study is known, and if we can know that how these transferritin molecules are forming there, you can tell little bit about the process what we considered as your biomineralization process. So, huge protein envelope is available, and you are supplying one after another from your food material the iron site, and that iron is going from one particular place, suppose your intestine or some other place, to the site where you have the ferritin. The ferritin molecules are not that it is distributed throughout your entire body. At some location it is there, and that location how you carry this. So, what you should know first, whether the molecules which are responsible for carrying these iron centers or how they are stored. So, these two are related. So, the transport as well as storage of these iron site, they are therefore related. How you can utilize those molecules? So, there will be some protein molecules which will be utilized for transferring your iron site from one point to the other. And once you transfer that, there something is available again which can store your metal center. So, that is there. So, one such molecule is ferritin and another is transferrin. So, name immediately tells us that this is responsible for your transfer of iron. So, that is why it is transferring, and your Fe is your iron, ferritin is also related to your iron site, and so these two are related to for your metal storage and structural ones. So, this will discussed next, and for completion of this particular transport and storage with respect to electron carrier, with respect to metal storage and structural, and the third one is O2 binding, that little bit we all know, this oxygen binding, and this oxygen binding is also related to your transport and storage. How you transfer oxygen dioxygen molecule and how you store your dioxygen molecule, because there we all know that we have this for O2 binding, we have the myoglobin molecule, we have the hemoglobin molecule, we have the hemerythrin molecule. And from the non-protein part, one part we will take, because for what we will discuss in possibly in the next class is our division from this non-protein part. One is your transport and structural, and this is very important, that we have moved from a protein part to a non-protein part. So, it may not be related to mammals or human system, it can be to your plant system or it can be to your bacterial system. How the bacteria is utilizing, because we all know that we are also all the time we should know little bit about this bacterial part of for their metal assimilation, because the bacterial infection always we are suffering. The pathogenic bacteria, how it can be controlled not only through medicine, but also from your balance for the metal ion. So, bacterial thing and all these things, they basically utilize the non-protein molecule for their transport and structural part related to your iron. So, that iron basically for this transport and structural part, we have molecules like siderophores. So, those siderophores, they are all iron bearing molecule. So, those iron bearing molecule, they give basically these siderophores, but these siderophore molecules are not protein molecules, they are small molecular weight molecules, and those molecules are utilized by the bacteria for transferring that iron from one side to the another. And to know the detail of these siderophores is very important, because they utilize some useful structure. So, when people determine the structure, people know the structure of all these molecules. The siderophore groups, they are basically very good ligand system, where one particular part is providing a bidentate part, and three of these groups are attached to a macrocyclic ring type of thing, and they are basically trapping that iron and transferring one from one part to the other. So, basically these things will see first, that the ferritin molecule, the transferrin molecule and the siderophores, and for that particular thing, and we will just simply go for that how these particular metal ions, how these metal ions play important role for some of our biochemical processes. So, most of these are coordination compounds involving one or more metal ions. Coordinated to organic groups of great size and complexity. So, we all be interested to know the function of these metal ions. So, for that particular thing, that you have the metal and the ligand system. So, when you have something that you have the ligand, it can also function as a bridge to two metal centers. So, the way we know that if you can have iron bound to water, at the same time you can have in the deprotonated form of the water, you get FeOH Fe system. So, for this particular purpose, when two such thing, that means when two of these interacting to this, forming a bridge system. So, you have the bridging group. So, how they are forming, because this iron is there, you have water coordinated, and we need to deprotonate this one particular water molecule, and one water molecule from the second iron center is leaving. So, that OH is giving you that bridge system. So, in a complex situation, what we can get further, that this OH group, when they are bound to your iron site, depending upon the available lone pair of electrons, because you have these water molecules, where we know that water molecules all have two lone pair of electrons, and those two lone pair of electrons can form two coordinate bonds. but the neutral water molecule is not strong enough to bridge that particular unit where you have two metal centers. So, if they go for deprotonation, and that deprotonation will help to provide you a charge on the group. So, is a uninegative charged species like that OH minus. Similarly, sometime what we find to know or detect any metal center which is bound to your hydroxide group can be very easily identified if you are able to find that okay, there are certain groups where iron is bound to your OH group. So, any spectroscopic technique which can be useful to identify this FeOH bond or FeOH Fe unit, which will be useful to know this particular motif. So, this particular motif can further be extended. So, when you remove this water molecule from here, another proton from this water molecule, you get the hydroxide group, and that hydroxide group with a charge have 3 lone pair of electrons. So, it can go basically for a triangular motif. So, in this particular case, this we are slowly moving towards to know that the how this ferritin assembly is taking place. So, if you have, because all the mineralization process also we know in a similar fashion the mineralization process is also taking place. That means, the hydroxide are formed first, and then hydroxides are transferring to the typical oxides, whether it is Fe2O3 or Fe3O4 type, and those hydroxides forming your corresponding oxides giving you the typical mineral. So, in the biomineralization process or during the ferritin formation, how these OH groups are useful to give you a huge cluster, because if this growth is continuing with OH, you can attach 3 iron centers, and if it is O2 minus, it can attach 4 Fe centers. So, is the another typical motif for the growth or for the assembly process, and those assembly processes what we are getting all these things from the simple ligand which is your water molecule. So, in this particular fashion that the bridging groups or the ligands can change the whole configuration. So, if this is 1, this is 2. So, binding to these negatively charged functional groups or ligands change the whole conformation of an enzyme or biomolecule. And during that process, what is happening there, that through coordination metal ion can enhance the acidity. Can't see. It's okay. Can enhance the acidity of coordinated molecule, which is very important. when iron is bound to your water molecule, we all know that this particular pK is changing. So, pK is getting lowered. So, you are getting some acidic water molecule attached to the iron center. So, that immediately goes for deprotonation. So, in the system itself, you are getting that iron center which is attached to the hydroxide, and when hydroxide is forming, starting from 1 iron center to 2 iron center to 3 iron center, and this hydroxide group is attached to 3 iron centers. So, pK is further lowered, and that particular one will undergo for further deprotonation, and you get a oxido-bridge system. So, that basically give you that how you bring 4 metal centers together, and that particular thing going further, because you have the other vacant sites from each and every iron site. So, that all these can also be occupied by water molecule or the hydroxide groups, and you can go for a corresponding polymerization process in that fashion. So, during this particular behavior, what we see that the metal plus ligand system, and in the biological system most of these ligands what will be getting from the proteins basically. From these proteins what we will get, and these amino acids, different amino acids say from this particular thiolate or cysteine, then you can have imidazole of histidine, carboxylate of glutamate and aspartate, and phenolate of tyrosinase. So, these are few examples for those groups, that when you have the ligand structure and those ligands are protein origin and you have some amino acids. So, all these polypeptide chains bearing those amino acids. So, always you have the amide backbone, and from those amide backbones you can have the pendant groups available, and those pendant groups like your cysteine, histidine, tyrosine, all these things they are dropping down from the backbone. So, depending upon the structure, suppose you have the corresponding primary structure, secondary structure or tertiary structure of the protein, you can have some useful donor groups available from sulfur, nitrogen, oxygen of the carboxylate group and the tyrosine from the phenolate unit. Those are available for binding the metal center. So, this particular thing, so in the next class we will see that how all these ligand systems and the metal center are useful for giving you a particular assembly. When we talk about the different ferritin molecules, how ferritin groups will be formed. So, you have a long polypeptide chain, and those polypeptide chains, whether they should be available for direct iron binding or not, that you should know first. So, from the very beginning of this classification of this metal and the ligand system from the biological origin, because the ligands are all from biological system and they are basically made up of the different amino acids. So, when they are coiled around to giving you a typical shape, and the available donor groups, if they are available from these long polypeptide chain, they will be coordinated to the metal center. And in other case, this particular one, what we find that devoid of the metal center, we get something which is the apoferritin part, when you do not have the metal center. So, this apoferritin part, and if we talk about the transferrin molecule, the apotransferrin part, how these protein structures are there and how these protein structures are changing through metal binding or metal coordination. So, that we will see from there that how it can grow from the typical point of view of your metal coordinating to your ligand system. Okay. Thank you.